Step outside tomorrow morning and hold your bare forearm in the light for a moment. You will feel warmth, and you will assume that warmth is the whole story, that sunlight stops at your skin, that it is a surface event, something that browns you or burns you and goes no deeper than the layer you can pinch between two fingers. That assumption is wrong by a margin that is almost difficult to believe. A specific fraction of the light landing on your arm is not stopping at the surface at all. It is passing through your skin, through the fat beneath it, sinking centimeters into the tissue, and arriving at structures so small that 10,000 of them would fit across the width of a single hair. And when it arrives, it does something to them. It changes how they work. It changes how much energy they make. The light reaches inside the engines of your cells, and it has been doing this every sunny day of your entire life without your permission and without your knowledge. The engines are your mitochondria. You have heard the phrase from a textbook, the powerhouse of the cell, and the phrase has been worn so smooth by repetition that it has stopped meaning anything. So let me make it mean something again. Inside almost every cell in your body sit hundreds, sometimes thousands of these structures, and their entire purpose is to take the food you eat and the oxygen you breathe and convert them into a molecule called ATP, adenosine triphosphate, the universal energy currency that every single process in your body runs on. Your heartbeat runs on ATP, your thoughts run on ATP, the repair of a wound, the firing of a neuron, the contraction of a muscle, all of it is paid for in this one molecule. You manufacture and burn through roughly your own body weight in ATP every single day. Not because you store it, you can only hold a few seconds worth at a time, but because your mitochondria remake it continuously, billions of times per second, in a process so relentless that if it stopped completely, you would be dead in seconds. Now here is where the light comes in. To make ATP, the mitochondrion runs what is called the electron transport chain, a series of 4 protein complexes embedded in its inner membrane, passing electrons down a line like a bucket brigade, using the energy released to pump protons and ultimately spin a molecular turbine. The final station in that chain, the 4th complex, is an enzyme called cytochrome C oxidase. And cytochrome C oxidase has an unusual property that almost nothing else in your body shares. It absorbs light. Specifically, it absorbs red and near-infrared light, wavelengths roughly between 600 and 900 nanometers, because it contains copper and iron centers that happen to soak up photons in exactly that band. This is not a metaphor, this is a measured optical property. You can shine red light on purified cytochrome C oxidase in a dish and watch its absorption spectrum respond. And red and near-infrared light is precisely the part of sunlight that penetrates tissue most deeply. Blue light scatters and stops near the surface. That is part of why the sky is blue, the short wavelengths bouncing everywhere. But the longer red and infrared wavelengths slip between your cells, scattering far less, reaching down through skin and into the muscle and the network of blood vessels below. So you have a light source, the sun, pouring out enormous quantities of exactly these wavelengths. You have tissue that lets those wavelengths through, and waiting at the bottom is an enzyme tuned by the chemistry of its metal atoms to absorb them. This is not a coincidence that biology stumbled into recently. This is a relationship as old as aerobic life itself. It helps to be precise about how deep the light actually goes, because the instinct is to assume the answer is fractions of a millimeter. It is not. Tissue has what optical physicists call a therapeutic window, a band of wavelengths between roughly 650 and 1200 nanometers, where two things that would otherwise stop light both happen to be weak at the same time. Below that band, the hemoglobin in your blood greedily absorbs the light. Above it, the water that makes up most of your body absorbs it. But in the gap between, in the red and near-infrared, hemoglobin has lost interest and water has not yet begun, and the light is free to travel. Measurements on living tissue put the meaningful penetration at 1 to 3 centimeters depending on the wavelength and the tissue. That is deep enough to reach the full thickness of your skin, the fat beneath it, the shallow muscle, and the dense network of capillaries threaded through all of it. A photon entering the back of your hand can plausibly reach bone. Your body is far more transparent to this light than your intuition allows. Hold a bright flashlight against your palm in a dark room, and the red glow that comes through the other side is not a trick. It is the therapeutic window made visible, the same physics that lets the sun's infrared reach the engines below. It is worth pausing on how that turbine actually works because the strangeness of it is what makes the light's effect so consequential. The machine that finally assembles ATP is called ATP synthase, and it is, in the most literal sense, a rotary motor, a molecular structure that physically spins like the rotor of an engine, driven by protons flooding back across the membrane through its core. Peter Mitchell proposed this in 1961, the idea that energy could be stored not as a chemical bond, but as a difference in proton concentration across a membrane, an electrical and chemical gradient holding potential the way water held behind a dam holds potential. He was ridiculed for years. The notion that the cell's energy was an electrical gradient rather than a tidy chemical reaction struck many biochemists as bizarre, and Mitchell, his health failing, funded part of his own research from a private laboratory in a restored manor house in Cornwall. In 1978, he received the Nobel Prize, and his chemiosmotic theory is now the bedrock of every cell biology course on Earth. The reason this matters here is that the entire gradient, the dam holding the water, is built by the electron transport chain, and cytochrome C oxidase is the chain's final and rate-limiting station. Speed up that one enzyme with light, and you raise the height of the dam. Everything downstream gets more powerful. The woman who first understood the light mechanism was Tina Karu, a biophysicist working in Russia from the 1980s onward. The phenomenon of light affecting cells had been noticed since the 1960s, almost by accident, when an early laser researcher named Undra Mester in Hungary tried to test whether laser light caused cancer in mice, and instead found the shaved fur on the treated mice grew back faster. For decades, this sat in a strange limbo, an effect people could measure but could not explain, which in science is almost worse than no effect at all, because without a mechanism, it looks like wishful thinking. Karu's contribution was to identify the receiver. She demonstrated through careful spectroscopy that the action spectrum of the biological response, the specific wavelengths that produced the strongest effect, matched the absorption spectrum of cytochrome C oxidase. The molecule that responds to the light is the same molecule that sits at the end of the energy chain. She had found the antenna. What does absorbing that photon actually do? Here the mechanism gets specific, and it gets strange. Inside a working mitochondrion, a certain amount of nitric oxide is always present, and it has a habit of binding to cytochrome C oxidase, sitting on the active site like a cork in a bottle, slowing the enzyme down. This is normal regulation. It is one of the ways the cell throttles its own energy production. When a photon of red light strikes the enzyme, it appears to knock that nitric oxide loose, dissociating it from the binding site. The cork comes out. And with the inhibitor gone, the electron transport chain speeds up. Oxygen is consumed faster, the proton gradient builds more steeply, and the molecular turbine spins harder. More ATP comes out the other end. A single photon, by displacing a single small molecule from a single enzyme, increases the energy output of the entire mitochondrion. This is the core of what is now called photobiomodulation, and it is the reason light therapy has moved from fringe curiosity to a subject of serious clinical research. But there is a second mechanism operating through the same enzyme at the same time, and it complicates the simple picture in a way the science is still working out. When the electron transport chain speeds up, it does not only make more ATP. It also produces a brief, controlled increase in what are called reactive oxygen species, molecules that in large quantities cause damage, the very molecules antioxidants are supposed to mop up. You would expect this to be bad. But in small, transient amounts, these molecules act as signals. They are read by the cell as information, triggering protective genetic programs, telling the cell to build more antioxidant defenses, to repair, to adapt. This is the same principle by which exercise, which floods your muscles with these molecules, ultimately makes you stronger rather than weaker. A small dose of stress that provokes a large protective response. The light is not just adding energy, it is sending a message. This dual effect is why clinical photobiomodulation produces results that sound at first almost too broad to be credible. The same red light has been shown to speed wound healing, reduce inflammation, ease joint pain, and accelerate recovery in damaged nerve tissue. When a single intervention seems to help a dozen unrelated conditions, the instinct of any careful person is suspicion, because that is exactly the shape of a fraud. But the breadth makes sense once you see the common root. These are not a dozen separate effects. They are one effect, restored mitochondrial output, expressed in a dozen different tissues. A wound heals faster because the cells rebuilding it have more energy to spend. Inflammation eases because the signaling molecules released by the light push cells out of an inflammatory state. The pathway is shared even when the symptom is not. NASA studied this for astronauts and for healing in microgravity. The United States military has studied it for traumatic brain injury, where the target is the energy-starved mitochondria of injured neurons. None of this requires believing anything mystical. It requires only believing that cells with more available energy do their jobs better, which is close to a tautology. There is a crucial catch, though, and it is the thing the supplement and gadget industry most wants you to forget. The dose-response curve for this effect is not a straight line. More light does not mean more benefit. It follows what biologists call a biphasic response, an idea that goes back to an old observation called the Arndt-Schulz rule. A small dose stimulates, a large dose inhibits, and the curve between them is shaped like a hill rather than a ramp. Push too much light into the tissue, and you do not supercharge the mitochondria. You overwhelm them, flooding the system with so many reactive oxygen species that the protective signal becomes genuine damage. The window of benefit is real but narrow, measured in minutes, not hours, and this is exactly why the sun has worked for your species for as long as it has existed. Morning sun delivers the right wavelengths at a moderate, slowly rising intensity, the body's natural dose. The problem of the modern world is not that we get too little of this exact light, it is that we get almost none of it in the form our biology was tuned to expect, and then nothing at all once we step indoors for the rest of the day. Now I want you to think about where in your body this matters most. Not your forearm, your eyes. The retina at the back of your eye is, by one measure, the most metabolically demanding tissue you possess. The photoreceptor cells burn through more energy for their size than almost any other cells in the body, and they are packed with mitochondria to pay for it. And this is where the work of Glenn Jeffrey, a neuroscientist at University College London, becomes genuinely startling. Jeffrey has spent years studying what near-infrared light does to aging retinas. In fruit flies, in mice, and then in human volunteers, he and his colleagues found that brief exposure to deep red light, around 670 nanometers, measurably improved the function of aging photoreceptors. In one study published in 2020, just a few minutes of 670 nanometer light per day improved color vision in people over 40, with the effect strongest in those whose vision had declined the most. The proposed reason is exactly the mechanism we have been describing. The light recharges the failing mitochondria of the retina, lifting their ATP output back toward youthful levels. And Jeffrey's later work pushed into something even stranger about timing. He found that the mitochondria of the retina are not equally responsive to light at all hours. Their sensitivity follows a daily rhythm, peaking in the morning. Light delivered in the morning produced a benefit. The same light delivered in the afternoon did much less, and at one point appeared to do nothing measurable at all. This suggests your mitochondria are not passive solar panels. They are clocks. They anticipate the light, they prepare for it. Which means the sun is not merely fuel arriving at random, it is a signal your cells have evolved to expect at a particular time of day, and the body has built its energy machinery around that expectation across hundreds of millions of years of mornings. Here is the layer the research is only beginning to map, and it is the one I find hardest to stop thinking about. For a long time we believed melatonin, the molecule famous for governing sleep, was made almost entirely in the pineal gland, a small structure deep in the brain, released into the blood at night to make you drowsy. That story is true, but radically incomplete. The work of Russell Reiter and colleagues at the University of Texas has built the case that the vast majority of the melatonin in your body is not made in the pineal gland at all. It is made inside your mitochondria, in nearly every cell, and it never enters the bloodstream. This mitochondrial melatonin is not a sleep signal. It is one of the most powerful antioxidants your cells produce, and it sits right where it is needed most, at the source of the reactive oxygen species, the inner membrane of the very engine that generates them. And here's the connection that, if it holds, reorganizes the whole picture. Reiter and others have proposed that the production of this mitochondrial melatonin is stimulated by near-infrared light, by the same red and infrared wavelengths that pour out of the sun and penetrate deep into your tissue, which would mean that daytime sunlight is doing two opposite-seeming things at once. The visible blue light entering your eyes is suppressing pineal melatonin, keeping you awake and alert. That part has been known for decades. But simultaneously, the infrared light soaking into your tissues may be loading your cells with subcellular melatonin, an internal antioxidant reserve produced exactly when the metabolic rate is highest and the need for protection is greatest. The sun would be revving your engines and protecting them in the same gesture. I have to be careful here because the evidence calibration matters. The cytochrome C oxidase mechanism is well established. The retinal benefits are demonstrated in controlled human trials, but the near-infrared stimulation of mitochondrial melatonin in living human tissue is still largely inferred from animal and cellular work. The physics is plausible. The foolproof in humans does not yet exist. This is science in the middle of refining itself, and it would be dishonest to pretend otherwise. But step back and consider what even the established parts imply about the modern world. We have spent roughly the last century moving indoors. We work under fluorescent tubes and LED panels, and we sit behind glass. And here's a detail almost no one knows. Ordinary window glass blocks very little visible light, but absorbs a significant portion of the infrared, and most modern energy-efficient glass is specifically engineered to block near-infrared, because that is how it keeps buildings cool. So the office worker who sits by a sunny window all day, feeling virtuous about the light, is bathed in the blue wavelengths that disrupt sleep while being filtered off from precisely the red and infrared wavelengths that may feed the mitochondria. We have built, without intending to, an environment that delivers the part of sunlight that agitates and removes the part that may restore. Our ancestors spent their days under open sky, under firelight at dusk. Firelight, notably, is extraordinarily rich in red and near-infrared. The spectrum our mitochondria evolved to expect is precisely the spectrum we have engineered out of our lives. Think about what that means for how tired so many people feel. Fatigue that no amount of sleep seems to fix, a low background hum of depletion. We reach for explanations in psychology or in screens or in diet, and those surely matter. But it is at least worth sitting with the possibility that some fraction of modern fatigue is, at the cellular level, an energy supply problem. Mitochondria running a little below their potential, because the daily infrared signal they evolved to receive has been quietly subtracted from the environment. Not a disease, a deficiency. The absence of an input the body assumed would always be there, the way it assumes there will be gravity, the way it assumes there will be morning. There is something almost poignant in the history here. Mester saw the mouse fur grow back in the 1960s and could not say why. Karu found the antenna in the 1980s and was, for years, working at the edges of mainstream acceptance, because light therapy sat too close to pseudoscience for comfortable company. Jeffrey's results were met with surprise verging on disbelief. The idea that you could improve human vision by shining a cheap red light into the eye for a few minutes sounds, on its face, like something sold in the back of a magazine. And yet the mechanism turned out to be real, rooted in the absorption spectrum of an enzyme that has been doing this since the first cells learned to breathe oxygen. The effect was never magic. It was physics, sitting unexplained for half a century because the receiver was too small to see and the idea was too simple to trust. So return to tomorrow morning, to your bare forearm held in the light. The warmth you feel is real, but it is the least interesting thing happening. Beneath that warmth, photons in the red and infrared are sliding through your skin, reaching enzymes tuned to catch them, knocking loose the molecular corks that throttle your energy production, and possibly loading your cells with an antioxidant made nowhere else and stored nowhere else. The sun is not warming you. The sun is reaching into the engines of trillions of cells and turning them up. It has been doing this every clear morning of your life. You simply could not feel the part that mattered. Stand in it tomorrow, not because it is pleasant, though it is, but because some part of you, far below the skin, has been waiting for that light since before you were born.